Hello, my name is Raj Dabulkar, roll number 7504, and the topic for my assignment is model organism. And the model organism that I have selected is Saccharomyces cerevisiae, commonly also known as yeast. So let's start with the presentation. So, what is the definition of model organism? Um, a model organism is a non-human species that is extensively studied in research laboratories for studying specific trait, diseases, or phenomena due to its short generation time, characteristic genome, or similarity to humans. A model organism is a species that has been widely been studied because it is easy to maintain and breed in a laboratory setting, and like it has a particular experimental advantage. Like when a scientist or researchers look for a model organism, they look for several traits like size, generation time, and its genetic manipulation, accessibility, and potential economical benefits. Apart from the one model organism that I have selected to present, there are others like E. coli, mouse, Coranohepatitis elegans, and Arabidopsis thaliana, and various other model organisms. So, on the basis of what characteristics a model organism is selected, the first is the genome similarity to humans. Like there should be some similarity between the model organism selected and human. Like genome similarity should be there, and genetic instability should not be there. Genetic instability means increased tendency for DNA mutations and other genetic changes. Genetic changes that occur during cell division, they should not be there. Can be grown in normal physiological conditions like normal temperature and pH. Availability of numerous genetic variants should be there. Accumulated body of knowledge about their genetic system should be there. Production of numerous progeny, ability to control genetic process in organism, and requirement of little space and resources as to easily maintain in lab. So, introduction to our model organism. The model organism is Saccharomyces cerevisiae, a species of yeast, which is unicellular and eukaryotic organism. It has been used in winemaking, baking, brewing since ancient times. Originally isolated from the skin of grapes, it is an important organism behind the most common type of fermentation. As we all know, all the fermentation is carried out by these yeast. It is one of the most extensively studied eukaryotic model organism in molecular biology, cell biology, and genetics. Saccharomyces cerevisiae cells are round, 5 to 10 micrometer in diameter. It produces by budding. Now let's see the classification of Saccharomyces cerevisiae. It belongs to kingdom fungi, phylum Ascomycota, class Hemiascomycetes, order Saccharomycetales, family Saccharomycetaceae, genus Saccharomyces, species Cerevisiae, and common name, as we all know, baker's yeast. Now let's look at the cell structure of the how a cell look of a unicellular fungi, that is yeast. It is a unicellular fungi. Cells are extremely variable in shape. This the one I have shown in the diagram is a oval shape, but they can also be globulose, elongated, or rectangular. Cells are hyaline. That means they are translucent or glassy in appearance, but in colonies they may appear white or cream colored or slightly brownish. Has a distinct cell wall which is made up of chitin and cell membrane enclosing granule cytoplasm. Cytoplasm is granule and enclosing large vacuoles and a nucleus. Also, several other cell organelles, like some cell organelles for reserve food materials, are present in cytoplasm, like oil, globules, glycogen, and volatile. This slide is important in context to why this model organism is selected. Number of chromosomes are 16 chromosomes in haploid spore. Number of genes are 6,275 genes. Genes common with humans are 3% of homologous genes to humans, and amount of DNA is 12 million base pair. The 23% of useful is useful in concept to knowing studying the gene function studies, and average gene size is 2000 base pair. Yeah, in May 1996, the complete genome of this organism was sequenced, and it was the first eukaryote to be whose genome was completely sequenced. Now let's see the life cycle of the Saccharomyces cerevisiae. Basically, there are two types of mating types: A type and alpha type. And when this A and alpha type come in contact under the influence of chemical signals, like pheromones, they secrete the chemical signals. They fuse to form a diploid cell, and this diploid cell undergoes its vegetative cycle, and simultaneously when there is like a nutrient rich condition, both the cycles like haploid and diploid cycle both proliferate equally by budding. But when there is a starvation, causes the diploid cells diploid cells 2n cells to function as ascus, and inside this ascus meiosis takes place, and 4 haploid ascospores are formed, as you can see at the top of the ppt, and for ascus containing 4 haploid ascospores. And at maturation, when this ascus breaks open, the A type is ascospores are released, and the A alpha mating type ascospores are released. And one point to note is that in this ascus, 2 are of A type and 2 are of alpha type ascospores, and they germinate individually, like, and carry out their haploid by budding, and carry out their haploid vegetative life cycle. And again, when they come in contact under the influence of chemical signals, the cycle continues. Like in basically in this diploid cell undergoes meiosis and forms haploid cells. Whereas haploid cells fuses to form a diploid cell. So it is also called as haplodiplobiontic life cycle, where both are equally dominant. And also, the diploid cells are heterozygous for mating type locus A and alpha, which means diploids are incapable of mating, whereas the haploid cells have either mating type A or mating type B, mating type alpha. So they are capable of mating. So now let's see the Saccharomyces cerevisiae as a model organism. A single cell organism is small with short generation time, doubling time 1.25 to 2 hours at 30 degrees Celsius, can be easily cultured. S. cerevisiae have nucleus containing chromosomes divided with meiosis, allowing it to be a candidate for sexual genetics research. S. cerevisiae can be transformed, allowing for either the addition of new genes or deletion to homologous recombination. Furthermore, the ability to grow S. cerevisiae as a haploid simplifies the creation of genetic knockout chains. As eukaryote, S. cerevisiae shares the complex internal cell structure of plant and animals without the high percentage of non-coding genes. It has less non-coding genes and also less junk DNA, and has a wealth of biological data that makes it attractive for as a model organism for study. And the advantage of using S. cerevisiae as a model organism are it is one of the long type classic model organism as the simplest eukaryote and the first eukaryote organism to be sequenced. Most essential cellular processes are conserved between yeast and humans. Yeast have simple nutritional requirement and can be easily grown in laboratory in standard lab condition, as it is one of the characteristic is important. And its genome contains 12 million base pair, 6,000 genes, as each share the same cell division process. Is invaluable for understanding loss of cell cycle control, and that occurs in cancerous cell, and so is well established for studies of cell division and cell cancer research. most striking feature is its existence as both haploid and diploid form, and that makes it easy to isolate recessive mutants in haploids. And by analyzing the S cerevisiae mutants observed from homologous recombination of foreign DNA, the functions of several proteins in vivo can be recognized. The entire genome of S. cerevisiae was sequenced in 1996, and since then it has been used as a eukaryotic model for studying protein interactions and infectious diseases. Its genome almost entirely free of introns. Its genome is free of introns. That makes it simple for cloning simple, and studying signaling proteins in yeast. As I have discussed during the cell life cycle, they secrete some pheromones or chemical signals, and this has advanced our understanding of brain and nervous system development. And also one more point is, it is a favorite model for studying cell cycling events during mitosis. This all points makes it the like a capable candidate for as a model organism. So yeah, thank you.